Okay, so today I will present some of the research done in my lab on the topic of explainable AI applied to biology and healthcare. So the work I'm about to present were done mostly by my AIMS lab students. It's a passionate researchers doing their PhD in computer science. Some of them are from the MSTP program pursuing computer science PhD, and then was funded mostly by NIH, NSF, and CZI, American Cancer Society. So we work on a broad spectrum of problems, including developing explainable AI techniques, and then also basic biology problems such as identifying cause and treatment of challenging diseases, and then also developing and then auditing clinical AI models. Okay, so today's talk will consist of two parts. In the first part, I will give you a high-level overview of many examples of what explainable AI can do to address new scientific questions in this space, biology and health, and providing some new ways to make biological discoveries, and then helping to make informed clinical decision and advance healthcare. And then hopefully I will convey the excitement of advancing AI and machine learning. The previous presenter, Caroline, emphasized, and then it was really great that, the importance of in this context of a causal inference, and it's really important to advance AI and machine learning and synergize that with the biomedical sciences. So in the second part, we are going to see how explainable AI can open new research directions in biology and healthcare, and which will give us a clue about how explainable AI needs to evolve and then also improve to solve a wide range of real-world problems in biology. Okay, so the key goal of explainable AI is to try to understand why a certain model output was generated. So say that we have a set of patient features and a trained machine learning model, we can predict various kinds of clinical outcomes. Let's say that we want to predict the kidney disease, ESRD in particular. However, in this traditional machine learning paradigms, you can't really answer a very natural question of why a certain prediction was made. And it is because with large modern data sets, the best accuracy is often achieved by complex models, even experts struggle to interpret. And then the idea of explainable AI is to explain the output of a complex black box machine learning model based on the contributions of input features for each individual prediction. So this black box machine learning model can represent supervised task, or it could also represent unsupervised tasks, such as representation learning, which Caroline talked a lot about in a previous talk. Say you have cell embeddings as output of this machine learning model. Say Y is a vector, and then you may want to know, what molecular drivers made certain cells or cell groups close to each other or distant to each other, or create certain patterns in the embedding space. So my lab has developed a series of explainable AI methods, and then the first one being named Shapley Additive Explanation. It's a general framework that can be applied to any model, and then also has a desirable theoretical properties, which other methods in this domain do not. So just to give you a very high-level basic idea of how it works, because that will tell you what the challenges are. The key question is how much adding a certain feature I changes the model output. So and then this may depend on which other features are also used by the model, subset of features. So then how about we consider all subsets of features S, and then average the effect of adding feature I across all subsets. So and then by doing that, the cooperative game theory guarantees us that the Shapley values to satisfy fairness axioms, which many other methods in this domain do not. Super intuitive, properties such as that, say feature I does not change the model output at all, regardless of which other features were used. Then you want this feature attribution, the phi value, to be 0. So such very intuitive properties. You want the method to be accurate and then satisfy all these properties. So turns out SHAP is the only way to get them. But, and developing SHAP, however, did not solve the problem, and then ended the research. It actually opened so many research directions in this explainable AI field, which really matters, which also matters a lot to biomedical sciences. So these key challenges involve proper feature removal. When I said, all considering all subsets of features, your machine learning models, in fact, you can't really have a subset of features, or you need to have all, values of all features. So, and then also, considering all subsets of features involve a serious computational complexity. So, and then our more recent work discussed that, and we showed that SHAP unifies a large portion of XAI literature with 25 methods. Many of them are actually used a lot in computational biology field. They follow basically the same basic principles, all explaining by removing features. And then this helps us to understand, what goes on when they fail, and then also improve and develop new XAI methods. And then on the computational side, we explored many avenues to make SHAP computation even feasible and faster. And then we developed, fast statistical estimation methods where we can detect convergence, and then another set of approaches for specific model types, such as tree models, tree ensembles, and then deep neural network, which matters a lot in the computational biology problems, and then more recently, transformers and graph neural networks. So, and then, using one of these developments, the tree SHAP, the method for tree ensemble. So using that method, we revealed individualized risk factors of chronic kidney disease, or all-cause mortalities, or other clinical conditions. So these results provide new insights, depending on what you predicted, biomedical insights or clinical insights in terms of what matters to those clinical conditions, or in biology, it will be phenotypes. So in a different study, we also showed that explainable prediction can improve clinicians' ability to predict events. For example, this ROC curve shows that the prediction performance for hypoxemia prediction, it means low blood oxygen level that can happen during the surgery when the patient is under anesthesia. It's a dangerous condition. And then our result implies that clinicians can detect millions of additional hypoxemic events annually with the aid of prediction with XAI capabilities. So in yet another study, we showed that XAI can help us interpret our biological age. So we named it Enable Age. In this study, here's how it works. So say that 65-year-old person who turned out to be 72 in terms of the biological age, and then we can obtain the explanations that increased or decreased the age, and then they come with the extent in the unit of years. So this is one of the desirable properties I described about the SHAP. So you will have the explanations will have exactly the same unit as what you predict. In this case, it will be biological age, so it's years. So obviously, cancer diagnosis accelerates biological age, and then also cysteine C is a kidney function measure. And then these explanations are provided for various mortality causes as well. And then we also perform the GWAS to identify potential genetic risks for age acceleration. So here the phenotype was biological age minus chronological age. So quickly, just to show you, what explainable AI can do in the field of Alzheimer's disease biology. Let's say we use a multitask deep neural network to accurately describe these complex relationships between 20,000 genes expression levels. The input layer would be 20,000 genes expression levels, and then say that you consider 6 AD neuropathological phenotypes, including CRAD score, plaque counts, and then A beta and then tau levels and so on. And then say we showed that this neural network more accurately predict this neuropathological phenotypes better than univariate model or linear models, but then the interpretability is an issue. Now with SHAP, we don't have to worry about that. We can compute individual genes contribution to each neuropathological phenotype through this complex black box model. So this in our study, this led to some unknown molecular mechanisms underlying the neuropathology. So we started to really, when we worked on this, project on this Alzheimer's disease and neuropathological phenotype, we started really thinking about further how to make the best use of XAI when you have a gene expression data or other kinds of omic data as input. Current XAI methods attempts to explain the prediction based on the input features. In this case, it would be 20,000 expression levels of genes. In fact, individual genes may not be the most interpretable unit, and then it may not be as interpretable as biological pathways. So some sort of functional unit, the set of genes that are known to be involved in the same biological processes. So we designed a new architecture and then developed a new XAI technique that basically organizes individual genes in the input layer into the pathways in this bottleneck middle layer. It's a set of genes again that are involved in the same biological processes. We use the reactome and the biocarta and the KEG. So that we also developed the principled ways based on SHAP to compute attributions to these pathways and then each pathway to individual genes as well. So this led us to an amazing finding that mitochondrial complex 1 is an important potential mediator for tolerance to beta toxicity. Basically, we had oxidative phosphorylation, one of the pathway in this bottleneck layer, which turned out to be really important. And then that's how we got from the gene expression data set from post-mortem brain. Some of them have Alzheimer's, some of them don't. And then we also the validation in vivo using A beta, the C. elegans that produces A beta in their body. So and then that tells us that it can there's a promising pharmacological avenues, and then XAI could identify the potential drug targets for Alzheimer's. So another idea to make the neural network architecture more interpretable. So for gene expression data set. Is to accommodate the target versus background setting. So especially single cell transcriptomic data sets are routinely collected in this manner. So in such a setting, it would be desirable to disentangle latent structures and the variations uniquely enriched in treated cells from those compared with the shared with the controls. So this led us to a new architecture to explicitly disentangle the data into shared and treatment specifics. We named the salient variables. So when we applied to this data to drug-treated cancer cells versus untreated cells, we see that the cells behave differently differently in the salient space. So this is treatment specific cells, depending on their p53 mutational status. So this is possible because the shared space absorb all these nuisance cell type specific signals. So we don't see this if we use the conventional methods that do not disentangle shared versus salient variables. This kind of finding is really important for personalized medicine, and it's known, and we are not saying that we discovered this, but it was a really good sanity check to see. You can see this differential responses between this p53 mutants and then versus not. So in the rest of this talk, I want to show you XAI is not just about interpreting models. It can do more than that in biomedicine. So it can even open new research directions. So for example, model auditing, and then cost-aware AI, but it doesn't have to be clinical. It can also be biological AI also. And then cancer therapy design for precision oncology. So briefly going over each of them. The first example is a model auditing framework for clinical AI, which revealed that basically many published clinical AI systems to detect COVID-19 from chest x-rays learned shortcuts rather than genuine pathology. So our results first illustrated a potentially dangerous condition in which the algorithms they appear highly accurate within one data set, even in a cross-validation setting. So that's red curve in these ROC curves, but fail when they tested in hospitals external to the training data. And to understand why these models fail to generalize, so we took a look at the saliency map, which is one form of the most popular form of explainable AI. It basically tells you which parts of the image were important to your AI system, AI model, prediction model. So for example, in this image, which turned out to be not COVID negative, it's right that the model used a clear long field as the basis to make that kind of decision, but it is not quite right that the model used the laterality marks. You see this left and right corner, top corners of this X-ray images. You certainly don't want your AI model to focus on that, that kind of information. And then also medical devices the patient is wearing to predict negative COVID-19 status, not based on genuine pathologies. So overall, throughout many XAI-based analysis, we basically stopped the field and moved to the wrong direction. There were already 6 published papers. Some of them are prestigious radiology journals, and hundreds of related models out there to learn the shortcut. We even found a startup in India who used some of these models to actually was going to use that in their country, and we sent a really nice email. The CEO replied really nicely saying that they are going to look into these issues. So we see that it's really important to dig into what an AI model was thinking. And then the next target is dermatology models. Dermatology AI models in collaboration with the Stanford Dermatology. So there are many cell phone apps, and then high-profile biological journal papers like including Nature. And then we saw similar concerning results. So it was consistent with what we saw in the COVID-19 project, where these models didn't work really well in terms of the accuracy. And then when we tried to interpret these models, we ran into an interesting challenge. So this is one example that still, explainable AI or computer science more broadly needs to be further improved to work, to solve real-world problems in biomedicine. So let's say you have this original image, so it's a dermoscopic image of the potential lesion, and it's predicted to be benign. But when you look at the saliency map, it's not really insightful, isn't it? It's basically pointing to all, the whole lesion. It doesn't really tell you what this, AI model, used. I mean, what aspect of this image led to that prediction. So, but if you modify the image and create a counterfactual, such that the model thinks that this is malignant, then you will see all these visual features, such as, I learned a lot of dermatology term in this project. So in the middle of the image, there is this bluish part, it's called blue-white veil, or, overall there is multiple colors, multicoloration that usually indicates melanoma. Then you can see that, this AI model, it was depending on this kind of, features. So we now, probably now it's in the Med archive, we submitted preprints that, that describes this method. So, and using, counterfactual explanations as a way to understand what AI model reasoning process is, and then also a way to automatically generate concept from images. So if you ask dermatologists to describe certain images, so it's either in the left or right, it will be really laborious for them to come up with all the medical concepts this image contains. And then we developed a foundational model approach that can give you with a text prompt, that this basically describes this image. So now we also, showed that XAI can help us to make an AI model most cost aware. Many, clinical AI literatures focus on getting prediction performance, and then biological AI literatures too. I mean, they celebrate good accuracies and that's it. However, if we really think about how these clinical AI systems are used, it's not hard to realize that many situations, gathering features is often costly, and that can lead to patient outcome. So say that you have a patient with a traumatic injury in front of you. This patient is bleeding, and then you want to predict whether this patient is going to have acute traumatic coagulopathy, ATC, and it's time sensitive. It requires a lot of blood, massive transfusion, and then so earlier transfusion leads to better outcomes, so less organ failure and so on. So faster diagnosis leads to faster treatment. So accurate prediction of ATC is great, but we really want this prediction to be fast. In this case, cost will be time. And then some of these features that need deeper examination aren't the ones you want to rely on too much. So our method, we named Co-AI, it aims to, to balance between the feature importance, which we can obtain feature importance levels using XAI methods, and then also cost the expert assessed, and to improve both of the cost efficiency and performance of a clinical prediction. So here is one example result where y-axis is the prediction accuracy, x-axis is the cost over the input features. So you can see Co-AI does better than existing clinical score, which is a PACT, P-A-C-T, and then you can see that Co-AI is just as accurate, but then in terms of the model cost, it's much shorter time needed to collect all the features, and then it's a general machine learning framework. You can improve, also existing clinical risk scores to predict the mortality at ICU, critically ill patients, and then we showed that the same way it outperforms all the other, the clinical index. So, and the last example of how explainable AI can help us open a new research direction. Let's talk about cancer combination therapy. The cutting edge in the treatment of cancer, like the blood cancer, acute myeloid leukemia, we focused on, has increasingly become combination therapy. And then the rationale there is, it makes a lot of sense. By choosing drugs that target complementary biological pathways, you can achieve greater anti-cancer efficacy. And then, but the issue is that choosing optimal combinations of drugs is a hard problem. There are already hundreds of individual FDA-approved drugs, which means that there are tens of thousands of possible pairwise combination. And then there could be more if you consider non-FDA approved drugs or combinations of more than 2 drugs. And then also, most importantly, different patients, even patients who have the same type of cancer, subtype of cancer, may respond differently to exact same drugs due to patient-specific genomic or expression characteristics. So when we have too many choices, it'll be extremely useful to know why or why not certain drug pairs are good options. But more than that, our goal is to understand the underlying principles of the biological mechanisms by which 2 drugs have a synergy for a particular patient. So we developed the Express framework, just recently published, and then we use collection of the data set with a bunch of samples, including characteristics of different patients' AML tumor. We use gene expression levels and the characteristics of a bunch of different drugs, combinations of drugs, such as structure of those drugs or biological targets, and they use this data to train a machine learning model to predict whether these 2 drugs are going to have a synergy or not. But more importantly, we do XAI analysis to understand which genes are important for synergy, and then also more broadly, which pathways. We also did the pathway level the importance computation to get a sense of which pathways are important. So just to give you the idea of our main result, we identified stemness, the genes that are involved in hematopoietic stem cell lineage, or some genes that are known to indicate how differentiated the cancer is, can be used as axes to design combination therapies. What that means is that when you consider 2 drugs, you better choose one drug that target more differentiated cancers, and then the other one being targeting less differentiated cancer. So it's a very brief, one-line result, but we did a ton of analysis to reach this, the conclusion, just to give you an idea of how explainable AI can go a lot further than merely predicting something accurately. So just to give you a, so I just show you a little bit of XAI today, explainable AI today. It's as I said, it's a non-trivial problem, still needs a lot of advancement in the field of machine learning and computer science. And then I also try to convey this excitement of synergizing between 2 goals, advancing AI and also advancing biology and health. And then I showed that, this CS or AI advances enables, and it drives the scientific interesting questions and clinical research in clinical medicine and also cancer biology, and then precision medicine, and also Alzheimer's disease, for which there aren't any drugs, long-term effective drugs, and so we aim to understand molecular basis of the disease. So that's it for today, and then I'll be really excited to discuss with you in the breakout discussion.